In Chapter 7, we take a look at cellular respiration. Cellular respiration is the process of breaking down a sugar molecule or other biological molecule in order to release ATP. Well, not necessarily to release it, but to produce ATP, to use the energy in the bonds of that molecule to make ATP. In Chapter 6, we learned about anabolic and catabolic reactions. Those are reactions that build molecules and break down molecules. And so we're going to dive into some of those specific reactions within a cell that involve the building and breaking of bonds and therefore the storing and releasing of energy. I'd strongly encourage you to watch this BioFlix video on cellular respiration. You can find it in the PowerPoint on Blackboard. You can also find it directly in the lecture materials. Students tend to find cellular respiration and photosynthesis a little bit overwhelming because there are a lot of steps, there are a lot of inputs, a lot of outputs, and just generally a lot of things to remember. So it might be helpful for you to watch it as a quick overview to sort of see what all we're getting into. Back in Chapter 1, we learned that all living organisms exchange energy and matter with their environment. Life requires energy in order to go, in order to perform all of its activities and all of its functions, and that energy is transferred throughout an ecosystem. If we look at this energy diagram here, this is showing the energy flow starting as light energy. So all of the energy on Earth ultimately comes from the sun, but that sun energy is not usable for chemical reactions. It's fine for warming things up and getting a suntan, but it's not good for running chemical reactions. And so that energy has to be converted into chemical energy. And that's what our photosynthetic organisms, such as our plants, cyanobacteria, do. They take in sunlight and they convert it into chemical energy at the most basic level in the form of a very simple sugar called G3P. Once that energy is put into a sugar molecule, it can be converted into other forms of molecules within the organism, or it can be used to release the energy in those bonds and produce ATP. It's the ATP that those organisms use, the chemical energy that they use to do work, things like growing, reproduction. All of those metabolic processes that we talked about very generally in Chapter 6. Ultimately, all of the energy works its way out of the ecosystem. It's transferred around and passed through. In the process, a lot of energy is lost as heat. So even as we're taking that sunlight energy and converting it into chemical energy, a lot of it is lost as heat. We also see chemical cycling diagram here. We're not going to talk about that too much, but I'll mention it since we have the image. Unlike the energy flow through an ecosystem, chemicals are actually cycled through. So we start here, chemicals in the soil are taken up by the plant. They are used, converted by the plant, eaten by consumers. So here we have that lovely caterpillar. So all of the chemicals that plant has that become food for that caterpillar are transferred into the caterpillar. Ultimately, that caterpillar, the plant, the mushrooms, everything that is using those chemicals from the soil and from each other are going to die, and decomposers are going to return those chemicals back into the soil, and the cycle starts over again. But as I said, we're going to focus on energy here in these chapters. So all of our energy transfer reactions come down to this process that links photosynthesis and cellular respiration. All of the metabolic reactions, all of the chemical reactions that govern living organisms are based on an organism's ability to transform the energy from the sun into chemical energy, or the energy ultimately produced by plants in the form of chemical energy, but the energy or the molecules that they consume must be broken down in order to release ATP. So we're going to talk again about anabolic and catabolic pathways, but most importantly in this chapter are catabolic pathways. The process of breaking down a sugar or other biological molecule in order to release energy and produce ATP is a catabolic one. Remember, a catabolic reaction is one that breaks bonds in chemical reactions. It breaks down molecules, and in doing so releases energy. That energy is then harnessed and loaded into ATP. Remember, ATP is our energy currency. It's the molecule that holds on to that energy in its phosphate bonds, and that phosphate is transferred in order to help do cellular work. So there are several processes by which energy is released. The one we're going to focus on is aerobic respiration. That's what we do. It's the use of oxygen to help break down glucose and produce ATP. We also have anaerobic respiration, which is a similar process to aerobic respiration, except that oxygen isn't used. And then we have fermentation, which is a type of energy transfer that organisms who will not use oxygen in the process of aerobic respiration, they can use another process called fermentation. But ultimately, all of the energy released by those three processes is used to make ATP. Before we can make ATP, though, we have to harness that energy from the sun. So on the right-hand side, we have our light energy entering the ecosystem. Photosynthetic organisms take in that light energy, convert it into organic molecules. We are generally going to use glucose as our organic molecule. We're going to say that glucose is the output of photosynthesis. It's actually a molecule called G3P, which is just a 3-carbon molecule that can be used to make a variety of other molecules. But in general, we're going to say glucose. That glucose is then transferred into the mitochondria in either the plant that has performed the photosynthesis or into the mitochondria of a consumer who has eaten the plant or a secondary consumer who's eaten a primary consumer. In any case, the biological molecules are taken into the mitochondria, broken down, and energy is produced. We also see a link between carbon dioxide and oxygen. One of the byproducts of photosynthesis is oxygen. So plants essentially breathe in carbon dioxide and breathe out oxygen, whereas we breathe in oxygen and breathe out carbon dioxide. So not only are organisms linked by the process of photosynthesis and the production of organic molecules that are used for ATP, but they're also linked by this input and output of carbon dioxide. However, the goal of cellular respiration is not the breathing in of oxygen and the releasing of carbon dioxide. The goal is the production of ATP. And that's something that students tend to get a little confused on, because we think of respiration, rightly so, as breathing. But the goal of cellular respiration is not breathing. That's the organism's goal. The goal of cellular respiration is the production of ATP. Let's take a look at mitochondria structure. We looked at this really briefly in chapter 4. Remember, the function of the mitochondria is energy production, and the structure of the mitochondria is very closely related to that function. We see it as a double membrane. It's got an outer membrane here and then an inner membrane here. The inner membrane is made up of these folded shapes called cristae. And cristae are folded because it increases the surface area. We talked about this as one of the ways that you can increase the surface area to volume ratio in a cell as a whole, but mitochondria also do that. By increasing the surface area of the inner membrane, those cristae have a larger surface area. And the process of cellular respiration actually occurs in those folded membranes of the mitochondria. And so by increasing the surface area, of those cristae, we allow more room for cellular respiration to occur. Some of the processes of cellular respiration actually occur embedded in that membrane. And so if we have more surface area, we can run the processes of cellular respiration more frequently. We also have this mitochondrial matrix, that's the area in between the cristae. We'll see that some of our cellular respiration occurs in there. And then it's important to note that the mitochondria have its own DNA. I think that we talked about this in chapter 4, but mitochondria has its own DNA, as does the chloroplast, and we'll talk about why that is in just a minute. Since we're on the structure of organelles, let's review chloroplast structure just really briefly. I'll do it again in chapter 8 when we actually get to when we actually get to photosynthesis. The chloroplast is the site of let me get on the right process here. The chloroplast is the site of photosynthesis, and photosynthesis occurs in a structure of the chloroplast called the thylakoid. The main component of the thylakoid is chlorophyll, which is our energy capturing molecule. If you look at those thylakoids, they are also highly folded. These little disc-like shapes are called the thylakoid, and the stack are called granum. Just like the cristae, these thylakoids are highly folded, and that's because it allows for increased surface area in which photosynthesis can occur in those thylakoid membranes. Just like the mitochondria, the chloroplast has a double membrane and also contains its own DNA. They both also, because they contain their own DNA, also contain ribosomes, which means that the chloroplast and the mitochondria can direct some of their own processes because of that DNA and also produce its own proteins. So they're both using their DNA and their ribosomes to produce molecular products specifically for photosynthesis or respiration. The reason that these two organelles have their own DNA is that it is theorized that they were originally their own cells that were engulfed by a sort of precursor eukaryotic cell. This is called the Endosymbiont Theory. So endo meaning inside, symbiont meaning a mutually beneficial relationship. So it is theorized that an ancestral eukaryotic cell first engulfed an oxygen-using bacterium, which became the mitochondria. After that, it sort of split off into two lines. One line continued on to our animal and fungi lineages, and the other one split off and engulfed a photosynthetic bacterium, which ultimately became the chloroplast. So here's a diagram that shows what I mean by all of this engulfing and symbiotic relationship. So we have this sort of precursor prokaryote, precursor to the eukaryotic cell. And at some point, it engulfed a non-photosynthetic prokaryote that could use oxygen to produce energy. When it did that, rather than digesting it and eating it, the two cells developed a mutually beneficial relationship with the precursor mitochondria and bacteria living inside of it. So this went on to form our animal and fungi and protist lineages. Excuse me. Remember that our protists are unicellular eukaryotes. So some of them went on to form that lineage. But one of them, at least one of them, went on and decided to engulf a photosynthetic bacterium. Again, this led to a symbiotic relationship and ultimately led to our plant lineage and some of our other photosynthetic cell lineages. We know that the mitochondria was engulfed first because all eukaryotic cells have mitochondria, whereas only some of them have chloroplasts. This is another way to show this endosymbiont theory. It looks a little complicated on the surface, but it's actually not too bad. We have our precursor cells here. This precursor cell has engulfed a precursor mitochondria that eventually evolved into that mitochondria. And so we have the origin of these organelle structures. Some of that line continued on to our animals, fungi, and protists, and at least one of them went on to engulf that chloroplast and became our photosynthetic protists in our plants. So let's begin talking about the breakdown of sugar into ATP. So in general, for this lecture, we're going to be talking about catabolism, so the breakdown of a molecule. All of the formulas in this chapter and in chapter 8 need to be memorized. Sorry. It's just how it is. I ask that my students memorize it because it helps them to keep track of what's going on in the process, what the ultimate goal of that process is. So we are mainly going to focus on aerobic respiration, and that is where oxygen is consumed as part of the process to produce ATP. So here we have glucose, that's our C6H12O6, our oxygen coming in. So if we're thinking about humans, we eat glucose, we breathe in oxygen. And in this process of producing ATP, we also release some carbon dioxide. So you all know we breathe out carbon dioxide, and we produce a little bit of water. And then also along with this reaction, we produce some heat. So that is how our bodies release heat. They really release heat from all of the chemical reactions that occur, but this reaction, the cellular respiration reaction, occurs so often that we're producing a lot of heat. The energy release from glucose is exergonic. So as we break down that glucose, we release energy to the amount of 686 kilocalories per mole of glucose. That might not mean anything to you. I'll just tell you that that's a lot of energy. In the process of anaerobic respiration, we follow the same pathway as aerobic respiration, breakdown of glucose, release of carbon dioxide and ATP, but instead of oxygen, there's another reactant, another molecule that is part of that process. And then finally, we have fermentation, which is a different process, a completely different process that does not use oxygen and is really only a partial breakdown of glucose. All right, so when we break down chemical bonds, we release energy, but we also relocate electrons. If you remember from chapter 2, we talked about electrons being in shells and electrons holding or containing a specific amount of energy. When you transfer an electron, you also transfer that energy with it. And so we're going to be talking about these energy or electron transfer reactions in this chapter. These are called oxidation reduction or redox reactions. And in these reactions, electrons are moved from one atom or one molecule to another. When a molecule is oxidized, or the process of oxidation occurs, there's a loss of electrons. When a molecule is reduced, or when the process of reduction occurs, a molecule is going to gain electrons. So an easy way to remember this is the term oil rig. You may have learned a slightly different one if you remember it from high school biology or another previous biology class. And you can use whichever little acronym allows you to remember what's going on. I like oil rig. Oil oxidation involves loss of electrons. Rig reduction involves gain of electrons. One thing that tends to confuse students, though, is when they see the charges on these molecules. So if you think about what's going on, oxidation is a loss of electrons, which means the negative charge is becoming more positive because the negatively charged electrons have left. So reduction actually refers to charge. When we say the term reduction, we mean that the charge is reduced. As a molecule loses electrons, it becomes more negative, which means the number that we see on the charge gets lower. It becomes more negative as well. So the charge is reduced. That's another way that you can remember that. So let's look at an example of a redox reaction. In this reaction, we have sodium and chloride. And remember, this is an ionic reaction where sodium transfers an electron to the chlorine. So here, the sodium loses an electron. Remember, sodium gives up its electron to the chlorine. So that means it is being oxidized. Oxidation involves loss of electrons. So we go from an uncharged sodium to a positively charged because it has lost an electron. When one atom loses an electron, the other must gain it. And so here we have chlorine, who is originally not charged, but remember, wants that electron. It wants to pull it towards it. And so when it does that, it gains an electron and the charge is reduced. So the chlorine in this situation is reduced. Another set of terms here, the oxidized substance is called the reducing agent. That means that it causes the reduction of the other molecule. So the sodium is oxidized and causes the reduction of chlorine. Conversely, the reduced substance is the oxidizing agent. So the substance that is reduced is causing the other to be oxidized. Remember, anytime you have a redox reaction, one atom has to gain an electron while the other one loses it. In these processes of redox reactions, you always have an energy transfer. And that's because, one, electrons have energy, but two, energy is required to move those electrons. Even though that sodium sort of willingly gives up its electron, you still need some energy to actually get it away. Redox reactions are really important in both cellular respiration and photosynthesis because there are a set of molecules called electron carriers. So we said that if you break down a bond and you release an electron, it can be gained by another atom. But that electron has to be moved somewhere. It just doesn't float over to the next step in the process. And it does that via these electron carriers. So for now, we're going to talk about an electron carrier called NADH. Remember, if one molecule is oxidized, the other is reduced. So let's look at this NAD molecule on the left. This is a complicated molecule. You don't have to know the structure, but you do have to know its function. And the function of this NAD molecule is to pick up electrons and move them somewhere else. So in the process, this NAD picks up or gains an electron. So remember, reduction involves gain. So this molecule is reduced. It picks up an electron, and as we'll see when we get to this whole process of cellular respiration, it's actually going to move that electron somewhere else. But the reduced form, the form of NAD plus that has gained an electron, is NADH. And the reason is that when it gains an electron, it also gains a hydrogen. In this case, actually 2 electrons. It gains 2 electrons, and along with it comes the hydrogen, because the hydrogen is actually the source of that electron that's required for the reduction. Remember, when electrons move, they take their energy with them. So the whole point of cellular respiration is to release the energy or the electrons from glucose and transfer that energy to ATP. That's the ultimate goal. And so we need these electron carriers to do that. We'll talk about several different electron shuttles. I tend to refer to them as shuttles rather than carriers. But The reason that we need these shuttles, specifically NAD plus for cellular respiration, is that we can't just release all of glucose's energy at once. If you just have a pile of glucose on a table and you light it on fire, you just light it on fire and release a whole bunch of heat and flames, which is really not useful inside the cell. And so NADH works to slowly remove the electrons from glucose, slowly take away its energy and transfer it to NADH and ultimately transfer it to ATP. So that's part of the reason that we need those electron carriers, is we need to slowly reduce the energy amount. We cannot just release it all at once. It would be too much for the cell.